Okay, so the next part of the discussion of the matter and energy unit is photosynthesis. Just talking to my son this weekend, come to find out, apparently on the MCAT, you don't even have to know about photosynthesis, which is quite odd because it's maybe arguably the most important chemical reaction on Earth, but why would you ever need to know that to represent biology? I don't know. Um, it turns out that photosynthesis rests primarily in a eukaryotic organism on the structure of the chloroplast. Turns out that many bacteria can do photosynthesis, and they have membranes inside, but generally they're not enclosed membranes, but they do carry out very similar chemical reactions. So let's get to it here. Let's see, let's select the ink. All right, so photosynthesis occurs in chloroplasts. We think chloroplasts probably descended from algae. And the outer envelope of a chloroplast has two membranes. So this is odd. We won't say much about this again, but these, where I'm pointing here, these two little dots represent the outer and then the inner part of the outer envelope of a chloroplast. There are actually three sets of membranes in a chloroplast. The outermost membrane, the inner membrane just inside of that, and then these membranes that are discs and layers on the inside. So inside of the chloroplast, there are membranes of another type. The membranes are arranged in discs called thylakoids. The thylakoids are usually in stacks. A stack of thylakoids is called a grana, or the stacks are called grana. An individual stack is called a granum. Basically, in the light, so when sunlight is striking these little discs, protons from here, that's called the stroma, the fluid inside the chloroplast, outside the thylakoids. Protons are pumped into the interior of these little discs. And the reason that is because the protons, which store energy, can be used to produce ATP. So in the light, the innermost spaces called the thylakoids serve as a proton reservoir, which is used for what we call the chemiosmotic synthesis of ATP. That is what I was talking about before in the previous lecture, just adding inorganic phosphate to ADP to make ATP. On these grana membranes, on the thylakoid membranes, an individual disc is called a thylakoid. A stack of them is called a grana. So on the thylakoid membranes, we have three basic molecules. Chlorophyll, which makes them green. And by the way, chlorophyll doesn't come in one variety. That would just be too easy. There are multiple. An electron transport chain. The electron transport chain's job is to pump the protons. And lastly, protein complexes that make the ATP. They're called ATP synthase or ATP synthetase. Both mean the same thing. The chloroplasts, we think, likely evolved from free-living photosynthetic bacteria. It turns out chloroplasts have their own DNA, and they have their own ribosomes for making a lot of their own proteins. So it turns out it looks like a long, long time ago the chloroplasts were free-living little bacteria, and they got captured by other bacteria maybe. This is around 2.5 billion years ago, and that arrangement has led to the trees that are outside of your house right now. That is a very successful arrangement. Symbiosis, it's a lot better sometimes to ask for help than to try to do everything, by yourself. So now the next thing we do is we go down and we talk briefly about these regions, the stroma and the thylakoids again. The stroma is the protein-rich fluid. That's this peach area that's around the thylakoids, but inside the double membrane. The stroma has the enzymes necessary to basically bond carbon dioxide into organic molecules. That's called carbon fixation. Fixation. Carbon fixation is bonding carbon dioxide to organic molecules. Carbon fixation actually happens inorganically. If you add organic acids to carbon dioxide, and just a few enzymes without any energy, you can get larger organic acids. The catch is, though, if you want to build anything worthwhile out of that, you need some ATP energy, and we'll talk more about that in a minute. We already mentioned chloroplasts have their own ribosomes, and most plant cells have at least 40 or more chloroplasts per cell. And before we dive in, the overall equation for photosynthesis, if you want to be a chemist about it, is 6 water molecules and 6 carbon dioxide molecules are assembled by this gadget up here into 1 glucose and 6 oxygen. And that's happening continuously in sunlight. So let's erase this stuff and let's go down into the nitty-gritty details. Now there's 2 kinds of electron flow in a chloroplast. There's non-cyclic and cyclic electron flow. I'm going to talk mostly about the non-cyclic electron flow, and I'll mention a few, later on, a little bit about cyclic electron flow. So there are 2 sets of reactions that happen in a chloroplast. The first set we're going to talk about is the light-dependent reactions. They're also called the light reactions, just light reaction. And there's 2 kinds of light reactions. There are non-cyclic electron transport light reactions, and there are cyclic electron transport light reactions. Right now we're going to talk about the non-cyclic. It turns out the chlorophylls that are on the thylakoid membranes are arranged in groups, hundreds of molecules actually, kind of like photo dishes. These are called photosystems. There are 2 general flavors of photosystems in the chloroplast. Flavor 1 is P700 and flavor 2 is P680. Those are the 2 important ones in this story. Basically, when light strikes a photosystem, one of the chlorophyll's electrons will be boosted to an escape velocity. It'll, like a bullet, hitting a target and blowing a piece of the target off and away. When a photon slams into a photosystem, an electron invariably is kicked right off of the photo dish or the photosystem, and a protein is waiting to catch it. That electron is not going to be wasted. A special electron-accepting protein is going to catch it. Those are usually called cytochromes. Now, the way it works, light, we have to talk about this in a sequence so that this makes some sense. Light striking photosystem 2 and 1 basically move electrons from water to an electron acceptor called NADP+. NADP+ is kind of like ATP in a way, except it's 2 ATPs stuck together, and they're not ATPs, they're ADPs, and there's a nitrogen-containing group attached to them. Now, it turns out that electron movement the way it works is when light strikes the photosystems, photosystem 2 is designed to throw electrons to photosystem 1. Photosystem 1 is designed to throw electrons to NADP+, and water is broken down to throw electrons to photosystem 2 so it can keep functioning. There's a really important enzyme called ATP synthase or ATP synthetase that's wedged in the thylakoid membrane that it's going to use the protons that are being pumped to make ATP. So let's go to this diagram down here and I'll explain how this pumping works. Let's see here. Now, the way the photosynthetic pathway works, the basic pathway, here's photosystem 2. Light hits it. An electron is energized. The electron is picked up by cytochromes. The cytochromes pass the electron from one to the next like a conga line until the electron finally gets to P700. These cytochromes here, as they're passing the electron along, they pump protons from the stroma to the interior of the thylakoid. I know that sounds ridiculous, but protons are really important in biology. They're what are allowing you to think right now. You use proton energy too. So the electrons going from photosystem 2 to photosystem 1 pass down a cytochrome chain, and you'll see the author try to draw it going downhill here, or the artist, and that's because this electron has high energy because the light hit it and energized it, and then it let its energy go and used it to pump a proton. Then when light hits P700, not necessarily the same electron, but an electron is energized to a high level, and then it comes down cytochromes. But this time it's not going to photosystem 2, it's going to NADP. And by going to NADP, it forms high energy hydrogen atoms. You might say, well, how does an electron become a hydrogen atom? Basically, all a hydrogen atom is a proton plus an electron. So NADP piggybacks a couple of protons with it. When the electrons from photosystem 1 come to it down the old cytochrome chain here, the electrons actually become parts of these protons and they become hydrogen atoms. Voila! We now have hydrogen, which is one of the essential ingredients of carbohydrate. It's not an accident. Glucose is C6H12O6. Lots of hydrogens in there, and here's where they're coming from. Meanwhile, back at the ranch, water molecules have to be broken down. This is not completely understood here, but there's a goofy manganese cytochrome that's involved with photosystem 2 here that basically somehow magically oxidizes water, pulls electrons off of it so that photosystem 2 can get replacement electrons for the ones that are getting jabbed off by the light that's hitting it. So what we have now is electrons coming from water, going to photosystem 2, energized, going down a cytochrome chain to photosystem 1, along the way pumping protons. Photosystem 1, electrons get energized, go down a cytochrome chain, and they end up on NADP. Now, that's the light reaction. The last thing I didn't mention, protons get made. The protons get made, I'm sorry. Yes, protons get pumped. Oxygen gas is made. I'm sorry, misstated that. Oxygen gas is made. This oxygen gas goes bubbling away. Blub, 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 blub. It goes bubbling out of the leaf or the chloroplast into the leaf air and goes out into space. Oxygen is as much as people hype it up, sometimes it's not as good as people say. Oxygen is an oxidizing agent. Believe it or not, if you breathe in pure oxygen for too long of a period of time, you get chemical pneumonia and you die because oxygen is actually a poison that is keeping you alive. You know how chemicals, when they're in the right amount, they're great, and when there's too much of them, they're often bad. Oxygen is one of those things. So the atmosphere is about 20% oxygen, which is A-OK for the human body. But 50 to 60% oxygen is more corrosive to you, and 100% oxygen can't be survived for too long. We found that out the hard way in the hospital. More on that at another time. So in summary, oh, by the way, the last thing I got to talk about here before we leave it, these protons are used by this really cool enzyme that is wedged in the thylakoid membrane. So if I draw a little thylakoid here, the enzyme's actually wedged in the membrane here, and it's got a little whirly-bird end on it. And this thing spins around, and it uses, so these protons are being pumped into this space here. This enzyme sucks these protons in, and out on the outside out here, it spins, and it actually takes in ADP and phosphate and cranks out ATP. We don't know exactly how it works, but it works. And that ATP is used in the dark reactions to actually make sugar. The two important products of the light reaction are ATP and the hydrogen atoms on NADPH. So in summary, non-cyclic electron flow in the thylakoid membrane stores light energy in two forms. The reducing potential of the hydrogen atoms, NADPH plus H plus, nitpicky point, one of the hydrogens is carried as an atom, the other one's carried as a proton. This thing's got 50 kilocalories per mole. That's a lot of energy. The second way that photosynthesis stores energy is it builds the proton gradient, which is used in turn to make good old ATP. So the ultimate products of the light reaction are this gadget and this gadget, ATP and NADPH plus H plus. Now, there are certain poisons that can actually block specific steps of poison. They'll block specific steps of photosynthesis. Paraquat is one of the most widely used herbicides in the world. It'll kill almost anything. It acts as an alternate electron acceptor between photosystem 1 and NADP plus. So then the plant can actually pass electrons to NADP, and as a result, it can't make NADPH. Without NADPH, it can't make sugar. The plant internally starves and dies. you may have never come into contact with paraquat. It's not used much here in the United States. Triazines, on the other hand, I guarantee you've ran into that. That's in almost every pool you've ever swam in. Triazines are common pool algicides. They block electron flow from plastoquinone to cytochrome B. That's right in there. So if you block the passage of electrons between photosystem 2 and photosystem 1, wow. Now it's like paraquat. You can't get any electrons over here because you've cut off this part of the circuit. And on top of it, you can't pump any protons, so you can't make any ATP. Ixnay on life for the plantae, so to speak. All right, so now let's talk about the dark reactions. The dark reactions don't really happen in the dark. We call them, I got to back up. I forgot to tell you about the cyclic electron flow. It turns out, small point, that photosystem 1 sometimes doesn't have anywhere to pass electrons to. Like it sometimes there's no NADP floating around to throw electrons on. So it can't actually shoot these electrons down here. So what happens is some of the electrons, sometimes from photosystem 1, instead of going to NADP, they jump back here and they go back down the chain. And can you see the advantage of throwing an electron back here and coming back down the chain? It's because they can pump protons in. So photosystem 1 is kind of impatient sometimes. If NADP is in short supply, which it usually is, we'll talk about that later, the protons end up still get pumped because the electron, when it gets energized, instead of going right turn, it goes, hey, Clyde, let's make a left. It goes left, jumps back onto the P2 to P1 chain, pumps a proton, comes back here, gets re-energized. If there's NADP available, it'll make a right turn. Otherwise, it makes a left. This is called cyclic electron flow. It only serves to keep the ADHD photosystem 1 busy, ultimately doing something which is pumping protons when it can't do what it wants to do, which is dump electrons onto NADP. All right, now I made a mess out of the screen, so I've got to keep moving here. All right, now the light-independent reactions, carbon fixation. Carbon fixation is kind of complicated, but we're only going to go over the important points here. There's lots of reactions that are summarized. You see all these like little arrows. These are all individual chemical reactions that would make your head spin and fall off. Mine too. So the light-independent reactions of photosynthesis are what we call actual carbon fixation. We also call those the dark reaction. I usually just call them dark reaction. That doesn't mean they happen in the dark. They will only happen in the light because they require hydrogen and ATP from the light reaction, and the light reaction can only make those in the light. So it turns out the dark reaction, a good place to start, is it sucks in CO2 and bonds it to a 5-carbon sugar called RUBP. So here's good old CO2 right here. And here's the good old RUBP is right here. I'm sorry, right here. You put these two together, and there's, see, 1, 2, 3, 4, 5. There's 5 little black balls. That's a 5-carbon sugar. This is a 1-carbon molecule. When you add a 5-carbon molecule to a 1-carbon molecule, you should make a 6-carbon molecule. And it does. The problem is the darn thing is unstable, and it forms 2 little 3-carbon molecules. These little 3-carbon molecules are called PGA. Okay? PGA is phosphoglycerate, or 3-PGA, 3-phosphoglycerate. 3-phosphoglycerate is a useful chemical, but it can't be used for anything really seriously awesome because it's got a carboxyl group on the end, which is really not a good biochemical group. Carboxyl groups look like this. They are a COOH cluster. So PGA is a 3-carbon acid, C, C, C. There's hydrogens around here with a phosphate sticking off and some hydrogens. But this carboxyl group on the end kind of sucks for the molecule. It's not a really good thing. So what does photosynthesis do? Take that carboxyl group off there. In the 2 most important chemical reactions that happen on good old planet Earth, the 3-phosphoglycerate is converted to G3P. G3P is, it doesn't have a carboxyl group on the end. It's got an aldehyde group on the end, and an aldehyde group is sort of awesome biochemically. So the aldehyde group is here's how the aldehyde group is achieved. By the way, here's the carboxyl group I'm talking about right there. C double bond O, single bond O right there. That's it. So first, an ATP is consumed. By doing that, you see the carboxyl group here? A phosphate is stuck on the end of that carboxyl group right there. It's stuck to it. Then the hydrogen of the NADP is used. That phosphate is ripped off and the hydrogen's right there. That's what's so awesome. This little chemical right here can be used to make anything. Amino acids, provided there's a nitrogen source. Nucleic acids need a phosphate and a nitrogen source. Any kind of carbohydrate, any kind of lipid can be made from G3P, one of the most important biochemicals on Earth. And G3P is not going to disappoint you here. G3P, in a series of chemical reactions, is used to make either sugar, I'll point that out, or it's recycled into more RUBP to do some more carbon dioxide capture in the dark reaction, which is a cycle. Some of the G3P is actually sent off to make good old glucose. Oops, I'm getting a messy diagram here. Some of the G3P is actually sent off to make glucose. The rest is recycled into more RUBP to capture more CO2. You might say, well, why doesn't the plant just turn all of the G3P into glucose? Okay, I could do that, but the problem is this is a 5-carbon sugar right here, which is like you eat lots of 5-carbon sugars every day, like fructose, and they're very energy, they're high in energy and they're hard to make, so the plant just recycles a bunch of this lovely G3P back into RUBP to keep capturing more CO2. Meanwhile, about 10% of the G3P that's made is kicked out and turned into good old glucose. And it's exported so the plant can burn the glucose with mitochondria, get the ATP out of it, and do the endergonic chemical reactions that got to happen in the cell. So let's go through the text here really fast just to make sure I didn't miss anything. It says CO2 reacts with a 5-carbon sugar called RUBP to form PGA. That's this is wrong. That's another error here. It's phosphoglycerate. The PGA, it says, then is converted to G3P by use of ATP and NADPH plus H plus from the light reactions. About 90% of the G3P is recycled back into more RUBP. 10% is converted to glucose. Here's a summary. RUBP plus CO2 plus these hydrogens and ATPs from the light reaction make more RUBP, some carbohydrate, and then NADPs and hydrogens to go back to the light reaction, so the photosystem 1 can dump electrons on here, and these go back for chemiosmosis to get put back together by the Whirlybird enzyme. It takes about 6 turns of the Calvin cycle to generate 1 glucose, and I talked about the ADP and the NADP getting recycled. There's a special enzyme that does this reaction right here. See where it says Rubisco? The reaction of RUBP with CO2, that's catalyzed by a reaction called Rubisco, by an enzyme called Rubisco. Rubisco is the most common protein on Earth. It's in like every algae and every plant on the planet. So it's the most ubiquitous protein worldwide on good old planet Earth. Beats every other protein out there. So the last look at photosynthesis here, before we go on, because the next thing is respiration, is the pathway of energy in photosynthesis involves the conversion of light energy to chemical energy. But let's take a look at the pathway now. The light energy turned into moving electron energy, which became hydrogen atom and proton pool energy, which became ATP energy, which became G3P stored chemical energy, which was then put into chemical energy of carbohydrates. I know that probably makes your head bleed, but that's the way it works. And if you read about it a few times, if you like biology, it's going to start sticking in your head. It's one of the coolest reaction sequences on good old planet Earth. Carbohydrates are really stable and can be stored for years. In an old house that's like in the Cathedral Notre Dame, or Notre Dame Cathedral in France that burned down, those logs, those timbers that were holding up the roof were put in place 1,000 years ago. And that energy, that's glucose that's in the wood. That's glucose in the form of cellulose. It's strung together in cellulose strings. It's extremely stable. It can last eons of time. On the other hand, ATP and proton pools, they only last milliseconds. They don't last a long time. Ultimately, what the plant did is it took fleeting light energy and put it into really stable long-term carbohydrate energy, which is really awesome. That way we got something to eat at lunch. So here's what we have. We have light shining on the photosystems. If you watch carefully here, it's about to end right now. So if we go back to the beginning, here we go. Here we start. Light hits photosystem 2. Electrons from water go to photosystem 2. From photosystem 2, they go to photosystem 1. In this transition, protons get pumped. The protons here are being used to do chemiosmosis. See, chemiosmosis makes ATP here. Meanwhile, photosystem 1 is dumping electrons onto NADPs. That is converting PGA to PGAL. The PGAL is recycled into more RUBP or turned into glucose. Woo! That was quick. Anyway, that concludes the photosynthesis part of the matter and energy story. More coming on respiration.